So now we have finished spinning our ammonium sulfate fractionation in our SS34 tubes. And you can see that we have a pellet right here and the supernatant. And the supernatant currently has 40% ammonium sulfate in it. So we're going to pour it into a graduated cylinder to determine the total volume of both of our tubes. I'd already poured one in. This is my second one. And you're going to read the volume just like you did last time, so that you know you have 60 mils, for example, and you're going to use that same ammonium sulfate table that you did. This time, we're starting at 40% on the left, and we're going across to 60% on the right-hand side. So this tells us 132 grams per liter of ammonium sulfate is what we need to add. So if we have 60 mils, 0.06 times 164, that tells me we need 9.84 grams of ammonium sulfate this time. So here we have 9.84 grams. We're going to, just like last time, have ice in the middle here. And again, I'm doing this just so you can see it. Started spinning with a stir bar in there on ice. And we're tapping our 9.84 grams slowly in there. When we finish, and again, over a 5-minute period, when we finish with our 9.84 grams of ammonium sulfate added in there, you will again see the solution become very cloudy. The cloudiness is because the proteins are precipitating out due to the ammonium sulfate. Now, I want to point out that this was a change in protocol. Your actual protocol tells you to go from 40% to 45% to 60%, but just to save time, we're going to go directly from 40 to 60% ammonium sulfate. We've tried it before. We know it does not hurt anything, and you don't get too many extra proteins or anything like that. So we're going to go ahead and save time by going directly from 40% to 60% ammonium sulfate. And then as soon as all of our ammonium sulfate has been added over that 5-minute period and allowed to slowly dissolve over that 5-minute period, we're going to again pour them into an SS34 tube, and we're going to again spin them at 14,000 RPM for 10 minutes. So we will again pick up when we're finished with that point. Okay, so this is the pellet that you see after your 60% ammonium sulfate fraction has been centrifuged. You have a pellet. Here you have a supernatant. Now the proteins in the supernatant are not proteins that you want, and your aldolase is now in the pellet. So we're going to pour off and get rid of the supernatant. So we have just the pellet. Then you're going to bring the pellet up in a total of, and remember we have 2 pellets because we had 2 SS34 tubes. You're going to bring them up in a total of 10 mils of resuspension buffer. So you can either do 10 mils on the first pellet and take all of it and then bring up the second pellet, or you can do 5 mils on each pellet. However, you know, assuming you have 2 pellets, however you want to do it. So basically, just like we resuspended before, pipetting up solution, putting the tip of our pipette in the pellet, and then squeezing out with some force, trying to get it all resuspended. So we're going to do that a couple times. So now we have 5 mils in one tube, 5 mils in the other tube. I'm simply combining all of our fractions in a single tube. After your column is all packed here, and you can see here I have buffer on the top of my column because I'm never ever going to let the column run dry. While you are putting buffer through your column, you can hook it up to a fraction collector. So you want to hook it up to the fraction collector. You're going to put tubes in the fraction collector. You will have a small piece of tubing that will go when the stopcock is open and allowing buffer to flow down through here. Will allow buffer to go through here, and you're going to put it in. You're going to put the end of your tubing into the little connector right here, and then that little connector will allow drop by drop to drop into the test tubes that you have set up here. Now it's important when setting it up, you need to make sure that this little wheel that's back here is completely covered by your fraction collector. So that's that little wheel right there. If you don't have this on the wheel properly, if you just have it on the wheel kind of like that, where the little teeth are not properly inside, your fraction collector will not be moving properly. So when you are setting up your fraction collector, this is very, very important to make sure that you have it set properly over those little teeth. Then you can set your fraction collector to any number of drops. In this case, I have it set to collect 40 drops before it moves to the next tube. If you want to advance it for any reason, oh, and here's how you set it drops. It'll do by time or it'll do by drops. We do by drops. So that's time and that's drops. If you want to advance it to the next tube, you hit advance and it moves forward to the next tube, starts over at 0, starts measuring how many drops have been put into that test tube. You want to start doing this with your very first column because you want to know, for the number of drops that you've selected, what is the volume of those drops? So if you have 40 drops, what is the volume of those 40 drops? And that's because you're going to be collecting all of your fractions by the number of drops, because we don't have anything else to measure volume on here. And with time, if you have more buffer, it'll flow faster or it'll flow slower. So time is not a good way to do it. So we're going to be using drops. So to be able to say equal volume in every single tube, if my particular protein comes out at tube number 10, and I know that I have 2 mils per tube, and it came out at tube number 10, that means it came out at 20 mils, because you're going to need to know how many, what volume each of your little fractions came out at. So the first thing that you're going to do on this column, you're going to remove all the excess buffer. Now remember, when I started talking, we had a bunch of excess buffer up here, which has slowly gone down. So now that excess buffer, there's very, very little of it in there. If your column has excess, you can simply take a little squeeze bulb pipette and remove the excess buffer. The first thing you're going to do is determine the void volume of your column. To find the void volume, you're going to use a chemical called blue dextran, which is incredibly large, molecular weight of around 2 million. And you're going to lay it on top of your column very carefully and very slowly. You're not going to be injecting it into the column at all. You want it in a very careful layer right over the top of your column material. So right on the top there. So one nice little band is, as straight as possible. My column's not quite straight. You can do a better column when you're doing it, but you basically want to have everything down at the bottom right there. Let me get this one drop in the side down. So now you'll see the blue dextran is making its way slowly into the column. You're going to let that blue dextran make its way slowly into the column, and before the column runs dry on the top, you're going to add 500 microliters or a mil of the gel equilibration buffer to the top of the column. So you're keeping buffer flowing through the column. You're never, ever letting your column run dry. But by the same token, if you add your buffer too soon, all you're doing is diluting out your blue dextran, and you don't want to do that. You want that blue dextran to be as concentrated as possible as it's moving down your column, because it's gonna spread out as it moves anyway. So you want that to be as tight as possible. So you're gonna keep an eye on your column. You never walk away when you're doing this. Never walk away, because that's when the column will run dry. And if the column runs dry, you are in trouble. Okay, so now all my blue dextran is in the column, and I'm adding some extra buffer on top. And as I add some extra regular buffer on top, now the blue dextran keeps moving down the column. The buffer then moves on top of it. And so what you'll see is you'll have your blue band running down the column, and you will have white beads on top of that. When you see those white beads on top of that, when your 500 microliters of material runs into the column, then and only then are you ready to add a greater amount of buffer to it. Now, remember, you have this whole time, you are collecting fractions in here, because the purpose of this is to find the void volume of the column. And you find the void volume of the column by the moment that you start adding things to the top of your column. You have started collecting fractions. Start on a new tube if you need to. And when you see the first blue drop come out the end of the tube, the first blue drop come out the end of the tube, that's when you're going to stop collecting. And at that point, when you've measured your first blue drop coming out, that is the void volume for your column. So I'm still watching this. I'm watching the buffer flow little by little into the column. So now you can see it's moved down the column, your band of blue dextran, and there are white beads on top. So when I add a little more buffer, I'm not going to be disturbing my beads. So now we're ready to add more buffer to the top of the column. And in your lab drawer, you will have an extra yellow funnel, a big yellow funnel, just like you saw me put on the first column. So you can take that big yellow funnel and put it up here, and you can even pour more buffer in. So basically, after you have begun this process, you can have a whole bunch of buffer up here, and then you can walk away. So once you have buffer up here, you can walk away until the blue is all the way down here, and you're ready to start watching it again and saying, okay, I'm going to be collecting my, fractions at this particular point. So now we're going to just watch that blue run down the column. So after your blue dextran has pretty much run through the column, and it's right around down here in terms of where you're seeing it, it does spread out some. You're going to use a piece of white paper behind it to sort of see and follow the progress of, okay, it looks a little bluer here, it looks a little bluer here as it comes down, and so it's looking a little bit blue to me, like coming right here. And so as you get that first drop of blue coming into your fraction collector, that's when you want to say, okay, stop the column. And then after you've done that, you can collect all of your fractions that you were collecting with 40 drops a piece in them, and measure the volume of those and say, okay, well, if this is 1 mil and I collected, say, 11 fractions until I saw that first blue drop, my void volume for this column is going to be 11 mils. Now you want to make sure you know your void volume fairly accurately, because if somebody has a little higher column or somebody has a little lower column, their void volume might be different. But the nice thing about knowing your void volume, when you are finding elution volume for your specifically sized proteins, you're going to be taking your elution volume for each of your known sizes of proteins and dividing by the void volume. And this will serve to equalize out any column, because it's a constant. for any single sized protein, your elution volume divided by your void volume. You could have a column that's twice as high, but twice as high would have twice the void volume. It would have twice the elution volume, but it also has twice the void volume, so that ratio will still be the same of elution volume divided by void volume. So you're finding, how many fractions you had in your column. Where did the first blue drop come out? What is the volume per fraction that I had in my column? And after you've done that, make sure you keep running your column so that you get all of the colored material, all of the blue dextran out of there. You don't want that to run over. So now you know your void volume for your column. The next things that you're going to be doing is taking proteins of known sizes. Refer back to my lecture for a picture of those proteins of known molecular weights. And you're going to be adding them just like I showed you on top of the column. The one with cytochrome C in it happens to be colored. The other ones are clear. So you need to make sure you have your technique down with the blue dextran and possibly using the cytochrome C before you try to do the ones that don't have color to them, because that the color in there lets you see how good of a job you were actually doing in terms of loading the column and spreading out your peak. Okay, so what you're going to do after you've determined your void volume, the next time you use your column, you're going to be loading proteins of known sizes. You can maybe have one protein in there, maybe you have two proteins in there of different sizes, and they're going to be separating out because this column separates by size. The reason the blue dextran is so useful, it's so large, 2 million molecular weight in here. And it's so large that it can't fit into any of those little nooks and crannies inside the column. So it's boom, going straight through. It'll go through your column before anything else will go through your column. So what you're doing is, for each of those proteins you're going to load, you're saying, relative to the void volume, the V0, how many mils did it take to elute my specific protein off, or the VE? And you're going to get that VE, V0 constant. And so you can be loading either one protein or two proteins. Your two proteins will separate as they go down the column. So they're going to start off in a single band up there, but as they move through the column, they start to separate more and more and more. So they're going to be in different tubes right here in your fraction collector. So when you're collecting, one fraction might be right here and another fraction might be right here. How are you going to know what fraction they're in? Well, remember in the protein purification lectures, we talked about using a spectrophotometer at A280 to be able to detect proteins. So you're going to use your handy dandy old spectrophotometer, and this time you're going to use a quartz cuvette. In the last experiment, you used a plastic cuvette. That was fine because we were basically using it at absorbance of 340, which was a visible wavelength. But this one's going to be an ultraviolet wavelength. UV radiation does not go through these plastic cuvettes. So you have to use a quartz cuvette, because the quartz cuvette will let the wavelength, the ultraviolet wavelength, go through so that you can accurately measure. So if you are having problems in your experiment of not being able to find where your protein is, they're all low, it's probably because you are not using a quartz cuvette. So you're going to use the gel equilibration buffer. You're going to blank in the spectrophotometer with gel equilibration buffer, and then one at a time you're going to, from the very start, your very first one. I started with tube number 5 actually, so I'm starting with tube number 5 because that's why I started collecting when I loaded my blue dextran. You're just going to pour some into your cuvette, nothing fancy. Pour it in. After, of course, you've blanked the spec with the gel equilibration buffer, and then put that into the spectrophotometer, get a reading, and then come back here and say, okay, now I know my reading for this particular protein, and I'm ready to do the next one. You can pour it out, and you can pour the next one in here. and read that one in there. So you can go down, you can go through here one at a time, pour them out. Just make sure you tap a little to get all the liquid out before you try to pour the next one in. So basically what you're going to end up with is, you know, low, 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 low. Oh, here's a peak. And maybe there's a little bit, but it's going lower, low, low, low. Oh, here's a peak. Okay. Well, if you have two proteins, you should have two absorbance peaks in there. And the elution volume for those two are going to be relative to the molecular weight. So you'll remember in the Aldolase Part 1 lecture, we talked about how if you have the larger molecular weight versus a small molecular weight on a gel exclusion column, the larger molecular weight comes out first. So if you have two peaks, then you have to say, well, of my two known sizes of proteins that I was adding, what were those two different sizes? So if you have something that's 160 kD versus something else that's 200 kD, okay, the 200 is larger, so that's going to come out before the 160, and something that's 160 is going to come out before something that is, you know, 100, or before something that is 40. So if you know what you're loading on there, and you should when you're calibrating your column, so that's what we're doing here, calibrating our column. And so you're going to be able to say, okay, if it's this size protein, this is going to be my VEV0, and you're going to be graphing that. Refer to the graph I showed you in the lecture and in your lab manual to see how that's going to look. So after you have done all this, remember you're also going to be putting your aldolase on here as a comparison. So you put your aldolase on here, and you're going to get a VE for aldolase. And when you do the VE divided by V0 for aldolase, then you're going to be able to put it on that graph, your calibration curve of your known sizes of proteins, VE divided by V0 for all the known sizes. And then you're going to be able to say, okay, here's my VE V0 for aldolase, and therefore here's the molecular weight for aldolase using that graph. So this is how we're going to be using this column. You're going to be loading multiple things over this column. And when you're finished with this column, at the very end of the experiment, you're going to put it back in the original bottle. So remember, you have multiple people in your group that are going to be able to move the experiment forward. So while one person may be working on your gel exclusion column, another person can be working on your phosphocellulose column. And the phosphocellulose beads will be prepared for you. So those phosphocellulose beads have a cellulose matrix and phosphates all over the outside. So remember in the theory lecture, you learned that the aldolase is binding the phosphocellulose beads two different ways. When we have our dialyzed sample of aldolase that we put over our PC beads, phosphocellulose has a charge attraction to those molecules of aldolase at the pH that we are using. So it starts off at pH 7.3. And we're going to be making this column, and we're going to be just simply pouring it. You need 15 mils of packed beads. So you're not going to be filling this one all the way up. You just need 15 mils of packed beads. How are you going to know where 15 mils is? Take a graduated cylinder, put 15 mils of water in it after having closed the spigot, and pour it in here, and then make a little mark on your column wherever the 15 mils is. So now you know what 15 mils needs to come up to. So you're going to take your beads that have been prepared for you, and you're going to simply pour them in your column. You're going to pour a little bit over because they're going to pack down. They'll pack down pretty quickly when you're pouring it. So after it finishes packing down, you're going to use your PC column buffer to wash all the beads off the sides here, and make sure that you have a nice packed column that is pH 7.3. So when you pour your aldolase onto this column, that's pH 7.3, it's binding to the column with a charge attraction and also as an affinity column. So this column is acting two different ways, with ion exchange and as an affinity column. So then we're going to be washing buffer through the column. And that wash buffer we're putting through, that's pH 7.8. So now we've changed the pH of this column. And so initially it's binding onto the phosphocellulose two different ways. And you can keep putting a little bit more as needed into your column. It's binding to the phosphocellulose two different ways. It's binding initially as an ion exchange column, and it's binding an affinity because the phosphate is a natural substrate. It fits in the same place that the fructose 1,6-bisphosphate fits into aldolase. So it's going to grab onto the column two different ways. So when you change that to pH 7.8, now you are losing its ability to function as an ion exchange column. And you're going to get a lot of proteins coming off with that wash buffer of pH 7.8. So you're going to keep putting buffer through, keep putting buffer through. You're going to have a real spike if you were to put this in a spectrophotometer, you'd see a really high absorbance because you have a lot of proteins coming off from where you've bound them to this column. But you don't even need to worry about reading the absorbance yet. So you're putting pH 7.8 in it, you're washing a bunch through. Eventually, after you've maybe put, say, 10, 15 mils of material of buffer through your column, of the pH 7.8 wash buffer through your column, then you're going to put a cuvette underneath it, and you're going to grab a sample in the cuvette. When your absorbance is less than 0.1, and so the absorbance may go up and then come back down, that's completely normal. When your absorbance is less than 0.1, then you're ready for the next step. And the next step in this column is washing off, is our elution buffer, washing off the aldolase from the column with the elution buffer. And to do that, it's going to be the same pH 7.8 buffer as you had been putting through, but now it's going to have fructose 1,6-bisphosphate in the buffer. And that fructose 1,6-bisphosphate is going to compete for the place where the aldolase is binding the phosphate on the phosphocellulose column. And so it likes the fructose 1,6-bisphosphate a little bit better than it likes the plain phosphate on the phosphocellulose column. So it's going to stick to the fructose 1,6-bisphosphate that's in solution, and it's going to come out of your column. So as soon as you add the elution buffer, then you need to start collecting fractions. Now, it's fairly easy to collect fractions even without a fraction collector doing it for you. If you have the fraction collector available, if your lab partner is not using it for the GE column, that's great. But maybe they are. So what you can do is you can put 2 mils, because you can collect 2 mil fractions here. You can put 2 mils into a test tube and make a little mark on it. And then all of these you can simply put underneath the column and collect fractions. So if this was 2 mils, you're going to collect this one until you have 2 mils. When you have 2 mils in here, you can switch columns and do 2 mils into the next one. So you can collect 2 mil, 2 mil, 2 mil, 2 mil fractions till maybe you have around 20 mils or so after you have eluted. Now, this is not your elution buffer. We're still pouring the column here. I'm describing what you're going to do in the future. So you can collect 2 mil fractions, and then you can take them and read the absorbance on them. So after you have collected maybe 10, 20 fractions, and you read the absorbance on them, you should have a really good peak with your aldolase. So usually it's around 2.5 on the spectrophotometer. Sometimes it's so high, you're out of range on the spectrophotometer. So it will say greater than 3. That's a good thing. You want that. You want to collect all your fractions, which are really, really high concentration. Basically, your 3 highest concentrations. Students ask me all the time, they say, well, I had 2 that they're over 3 and I had 1 that was 1.5. How many should I collect? It's like, well, if you have 2 that are really good, then you can just collect 2. But if you have If you have 3 that are really good, that's good too. And then you're going to pool them all together. Now, we're almost through with this column, so I'm going to tell you, when you load, when you have your aldolase that's been dialyzed and you're getting ready to add it to your column, before you add your aldolase, your dialyzed sample of aldolase to this column, you want to save a sample. Remember, we saved a sample earlier. That was what we called S1. We did that after we broke open all of our cells, so we have the total protein that we started out with. And then we ammonium sulfate fractionated our proteins. And so the ammonium sulfate fractionation of our proteins, we have fewer proteins now. We got rid of a lot of them. And then we dialyzed. So before you put it on the column here, you want to save a 1 ml sample of it. That's going to be your S2. After you put the pH 7.8 solution to get rid of all the excess proteins, and when you use the elution buffer to specifically elute just your aldolase off of the column, your peak that you've pooled, your 3 highest, you're going to mix together, and that's going to be your S3. So it's very important to be saving samples. Don't forget to save samples as you go along. You're going to want them, because remember, in the protein purification lecture, I talked about how you were going to fill out a chart. So you're going to be filling out a chart for me to measure your progress. You're going to be measuring the activity of aldolase for S1, S2, S3, S4. You're going to be measuring protein concentration for S1, S2, S3, S4. You're going to be comparing them. So, and you're going to be saying, well, how were we able to increase the specificity of our enzyme? So is our specific activity increasing? Is it more pure, basically, is what you're going to be answering at the end of this lab. After you have the aldolase, that's your S3 coming off this column, that's the aldolase that you're going to be loading onto the sizing column. After it's purified here, you're going to be loading it onto the sizing column there and measuring the size of it. And as it's eluted, that is your S4 that comes off. So don't throw away your aldolase after you've put it either on this column or this column. Make sure to save it. You don't have to save all your protein standards that you're going to be putting through, but you do need to save the aldolase that you're putting through. And we'll talk more about the next steps. And now we need to dialyze. We need to get rid of the ammonium sulfate, because right now, as long as there's 60% ammonium sulfate in here, if we were to spin this down again, we would simply be pelleting our protein out again, because it's not yet really in solution. It's been suspended in solution, but it is not yet solubilized in solution. So to do this, we're going to put on gloves. So you have to make sure you are wearing gloves before you touch dialysis tubing. So this is dialysis tubing, and there'll be a large jar of it, and you're going to just take some water, and you're going to open up the dialysis tubing and squirt water down inside the dialysis tubing to rinse it all off. And then between your fingers, squeezing it all down like that. And you're going to put a dialysis clip. Now we have 2 different types of dialysis clips. This one is weighted. So you're going to put the weighted one on the bottom and clip it securely. So you want to make sure it's securely clipped. If you're doing this with a number of groups, you would want to put some kind of a label on there, maybe with your marking pen. And then you need to put your solution inside the dialysis bag. So now we're going to pipette out our solution and place it in the dialysis bag. Make sure your tip is in the bag before you start pipetting it out. You should see it run into the dialysis bag and start filling your dialysis bag. So now we have pipetted our entire sample out into the dialysis bag, and we're going to put a second dialysis clip on here. We're going to leave a little bit of space at the top of the bag to allow for expansion of the solution. We're going to clip it firmly at the top, and we're going to put it into a beaker where we have our resuspension buffer. And over the next minimum 4 hours with buffer changes, it's all the ammonium sulfate is going to leave the dialysis bag. All of the solution is going to come in. Now notice we have a buffer change. We had phosphate buffer previously that we had used to bring up our solution in, and now we're going to have tris buffer. So we're allowing the ammonium sulfate and the phosphate to leave the dialysis bag, and we're allowing the tris to come into the dialysis bag. Now, because you're going to have a certain amount of ammonium sulfate, you want to try to minimize it, you're going to dialyze, and we have a little stir bar down there. So we're going to be slowly spinning the solution. It's a very slow spin of the solution, and we're going to allow it to dialyze for 2 hours. We're going to remove the buffer, and then we're going to put more of the same buffer on there and allow it to dialyze for a minimum of 2 more hours. For purposes of your class, we're just going to do until the next class period. But if you were working in a research lab, a minimum of 2 hours, change the buffer. 2 hours is what people normally do, although longer is fine.